Hello everyone, welcome to our new lecture. We are learning chapter number 10, that is cell and cell organelles. In our previous lecture, we have studied one of the important cell organelle, and that was nucleus. Now we are going to discuss the other cell organelles, okay, which are again important for the cell, and those organelles are endoplasmic reticulum. Then we will see the Golgi complex. Then we are going to discuss about lysosomes and their functions. Then also we will study about mitochondria and so on. Okay, now let us see what is endoplasmic reticulum. Now the organelle which conducts various substances inside the cell is called endoplasmic reticulum. So this is the organelle which is going to conduct various substances, okay, from the outside to the inside of a cell, okay, and that organelle is called as endoplasmic reticulum. Okay, so the function is it is going to take the important substances from the surrounding, that is from the liquid blood to the cell. Okay, the endoplasmic reticulum has a net like structure. So the structure is like a net consisting of interconnected miniature tubes and sheets filled with fluid. Okay, so you will see that lots of tube like structures are there. Okay, tube like structures, you can see those tube like structures in the given figure. Okay, and all these tube like structures are filled with a fluid. Okay, endoplasmic reticulum, that is ER, is connected to nucleus from inner side to plasma membrane from outer side. Okay, so this endoplasmic reticulum, it is connected to nucleus from the inner side, and then it is connected to a plasma membrane from the outer side. Okay, the rough endoplasmic reticulum, that is rough ER, has ribosome granules on its outer surface. So this particular endoplasmic reticulum have ribosomal granules, okay, on the outer surface of it. Now what are the functions of ER? There are three functions mentioned here. It is framework that supports the cell. Okay, it is the framework which is going to support the cell. Then the conduction of proteins, okay, as we have seen in the definition itself, that it conducts various substances inside the cell, so it also conducts the protein within the cell. Okay, then third function is that toxins that have entered the body through food, air and water are made water soluble by ER and then flushed out of the body. So suppose if any kind of toxin or any kind of unwanted material has entered the cell, okay, via food, air or water, then that unwanted material or toxin will be eliminated by the endoplasmic reticulum. And how it will be eliminated? The toxin is going to be mixed with the water. Okay, that means it is going to dissolve in the water, and that water will be flushed out of the body or of the cell. Okay, so these are the three functions of endoplasmic reticulum. Now let us see the another important cell organelle, and that is Golgi complex. Now it is made of 5 to 8 hollow and flat sacs placed parallel to each other. Okay, the sac, okay, it is made up of 5 to 8 hollow flat like structure which are called as sacs. Okay, they are placed parallel to each other. So you will see sac 1, sac 2, sac 3, sac 4, sac 5 and sac 6. Okay, so all these sacs are placed parallel to each other. Okay, these sacs are called cisternae. Okay, if we want to consider one of the part or one of the parallel sacs, okay, then we are going to call it as cisternae. Okay, and are filled with different enzymes. So there are different enzymes, okay, which are responsible for the growth of a cell or the body. Okay, those enzymes are there within the sacs or cisternae. Okay, the proteins coming from ER are enclosed in the vesicles which come towards Golgi complex via cytoplasm. So these vesicles, okay, small structure, circular structure that you are seeing here. Okay, these structures store the proteins, and these proteins are enclosed in a vesicle. Okay, this spherical structure, and this vesicle is going to be incorporated with the Golgi complex. Okay, Golgi complex and the vesicles are going to be joined together. Okay, and this vesicle is going towards the Golgi complex via cytoplasm. They fuse with the formation phase of the Golgi membranes and empty their contents in the cisternae. So these vesicles are going to take the proteins, okay, and then they are going to provide the protein to the Golgi complex. Okay, they are going to empty their contents. That means they are going to empty their proteins in the cisternae. As they pass through the cisternae, they are chemically modified with the help of enzymes. Okay, then they are again packed in the vesicles. These vesicles exit from the maturation phase. The cisternae work like a packing department that packs and distributes substances. So what is a cisternae? It is a part of the cell or part of the Golgi complex which acts like a packing department. Okay, so what it does, it packs some substances and then it distributes that substance to the other parts of the cell. The functions are: Golgi complex is the secretory organ of the cell. Okay, it is considered as the secretory of organ of the cell. That means it is going to secrete important enzyme. Okay, then it modifies, sorts and packs materials synthesized in the cell, that is enzymes, mucus, proteins, pigments. So whatever material which is synthesized within the cell, okay, it is modified, sorted and packed by the cisternae or by the Golgi complex and dispatches them to various targets like plasma membrane, lysosome, etc. So in short, we can say that Golgi complex is like a factory which distributes the product. Okay, it produces vacuoles and secretory vesicles. Okay, this is the third function of the Golgi complex. Then fourth function, it helps in the formation of cell wall, plasma membrane and lysosomes. So this is the fourth function, okay, wherein we have seen that Golgi complex is going to help in the formation of cell wall, plasma membrane and lysosomes. Okay, now the name Golgi complex, it is named after one of the scientists and whose name is Camillo Golgi. Okay, he was the one who developed the staining technique called black reaction, and with the help of it he studied nervous system. Okay, so this scientist had studied the nervous system, and to his honor the name Golgi complex has been given. He won the Nobel Prize of 1906 along with Santiago Cajal for the study of structure of nervous system. Okay, so for his studies in the field of nervous system or neurology, he has been awarded with the Nobel Prize also in the year 1906. Now let us see the lysosomes and its function. Now, due to various metabolic activities in the cell, organic waste is generated and lysosomes digest the waste. Okay, so lots of metabolic activities are taking place within the cell, and definitely when some work is performed or some reactions are taking place, we will get a byproduct, and that byproduct which is generated within the cell is called as organic waste. Okay, now the function of the lysosome is that it is going to digest that waste. Okay, so lysosomes are simple single membrane bound sacs filled with digestive enzymes. So these digestive enzymes are going to digest the organic waste generated due to metabolic activities performed within the cell. Okay, you can see the structure of lysosome over here. The single membrane is there. Okay, and inside the membrane you will see an enzyme complex. Now the functions of lysosomes are, okay, it is used, okay, in immune system where it destroys viruses and bacteria that attack the cell. Then the second function is demolition squads. It is destroys worn out cellular organelles and organic debris. So if any of the cell which is died or which is in not use, okay, or which is not required, those cells are going to be destroyed by the lysosome itself. Okay, and organic waste is also destroyed by the lysosome. Now this destruction of organic debris is called as autolysis. Then third is suicide bags. Okay, when a cell becomes old or is damaged, lysosomes burst and enzymes digest their own cells. Okay, so if a particular cell is not in function, or it has been old, or it is damaged for some reason, then this particular organelle lysosome, it is going to be burst out. Okay, there will be a destruction in the membrane and the enzymes will digest the own cell. Okay, then fourth function is during starvation lysosomes digest stored proteins, fats. Okay, now if some amount of protein is stored, or if it is some amount of protein or fat which is stored, then during the period of starvation, when the required amount of protein is not there with the cell, then lysosome is going to digest it. Okay, so this was the fourth function of lysosomes. Okay, now next is mitochondria. So each cell requires energy, and this energy is produced in the mitochondria. We all know that cell is performing a work, and to perform any work we need energy. So cell also requires energy, and the energy which is utilized by the cell is produced in the organelle called as mitochondria. Okay, which is a very important organelle of the cell. Okay, under the electron microscope, a mitochondrion is seen as a double membrane structure. Okay, if you observe the mitochondrion under an electron microscope, you will see that this organelle is a double membrane structure. Okay, there are two membranes, inner membrane and outer membrane. Okay, the outer membrane is porous and the inner membrane is deeply folded. Okay, these folds are called cristae. So the folds of the inner membrane, okay, it is called as cristae. The inner cavity is filled with proteinaceous gel-like matrix containing ribosomes, phosphate granules and DNA. So inside the mitochondria, within the inner membrane, you will see some important inner cavity. Okay, inner cavity of this inner membrane is filled with ribosomes, phosphate granules and DNA. Okay, that is deoxyribonucleic acid. Therefore, it can produce its own proteins. Okay, mitochondria is capable of producing its own proteins. With the help of enzymes, mitochondria oxidize carbohydrates and fats in the cell. The energy released in this process is stored in the mitochondria in the form of ATP. So whatever energy which will be generated within the mitochondria is going to be stored in the form of ATP, and the full form of ATP is adenosine triphosphate. Okay, very important full form, very important abbreviation, that ATP stands for adenosine triphosphate, which is generated when the energy is released, okay, in the mitochondria. Now plant cells have less mitochondria than animal cells. Keep this in mind, that the amount of mitochondria in plant cell is less than the animal cells. Now the functions of mitochondria is to produce energy rich compound, that is ATP. So this is the main function of mitochondria. It will produce ATPs, okay, that is adenosine triphosphate. Then synthesis of proteins, carbohydrates, lipids, etc. by using the energy in ATP. Second function is that it is going to synthesize protein, carbohydrate, lipids, okay, all these important nutrient molecules, okay, and it will be produced by using the energy which is generated in the form of ATP. Okay, now we have to keep in mind that mitochondria are absent in RBCs, so you will not see mitochondria, okay, this organelle, in the red blood corpuscles. Due to this, the oxygen which is carried by them is not used for themselves. So it is very important, because if mitochondria will have been present in the RBCs, then this particular organelle will have used the oxygen, okay, which is transported to the other cells via blood. Okay, now next organelle is vacuoles. Okay, so vacuoles are the storage sacs for solid or liquid contains. They don't have any typical shape or size. The structure of the vacuole changes according to the need of the cell. Vacuole is bound by single membrane. Okay, we have to keep this in mind. that vacuoles are nothing but a storage sacs, okay, and in this sacs you will find some solid or liquid content. Now, vacuoles do not have a typical shape or size. That means they do not have a well-defined structure. The structure of the vacuole changes, okay, as per the need of the cell. And we have to also keep this in mind that vacuole have only single membrane. Okay, it does not have double membrane or triple membranes. It only have single membrane. Now the functions of vacuole are to maintain the osmotic pressure of the cell. This is the first function. It is going to maintain the osmotic pressure, okay, within the cell. To store metabolic byproducts and end products, okay. So whatever byproducts which is generated, it will be stored by this particular organelle called as vacuole, and it will also store the end products, okay. Suppose if energy is required of 40 joule energy is required and generated energy is 50 joule, then definitely 10 joule is extra, and that extra 10 joule will be stored by the vacuole, okay, and which can be used in near future. In animal cell, they store waste products and food, while in the amoeba, it stores food before digestion. Okay, so in animal cell, you will see that the waste product or the food, okay, which is there in the animal, it is stored, okay, by this particular organelle called as vacuole. And if we talk about amoeba, which is a unicellular bacteria, okay, in that the digestion process, okay, before the digestion process starts, the food is stored by the vacuole. Fourth function is in plant cell, vacuoles are full of cell sap and provide turgidity, rigidity to them. Okay, so as in a plant cell, you will see that all the vacuoles have a full cell sap. Okay, so due to that, these two properties are indulged within the vacuole of a cell, that is turgidity and rigidity. Now the next organelle is plastid. Why are plant leaves green and flowers red? Okay, this question, a very important question. Why the leaves of the plants are green in color? The reason behind it is that the important pigment, that is chloroplast, which is there in the plant cell, okay, and due to that pigment, the color of the leaves is definitely green in nature. Okay, as the pigment also have a green color. Now here some color of the plant is given and the pigment which is found in those plants. So as I have said, green leaves will definitely have this pigment that is called as chlorophyll, and the orange leaf will have a carotene pigment. Then yellow have a xanthophyll, dark purple have anthocyanin, and the dark pink, okay, this particular plant have a betalains as a pigment. Okay, now let us see what is leucoplast. So leucoplast are nothing but white or colorless plastids. Okay, so some plastids which are white in color or colorless, they can be used in leucoplast. Then chromoplast are colored plastid. Okay, so different colors of the plastid. Okay, they are nothing but chromoplast. Chloroplast can be converted into other types of chromoplast. Example, raw green tomatoes turn red due to breakdown of chlorophyll and synthesis of lycopene. So definitely it is tough to convert the chromoplast to chloroplast or leucoplast to chloroplast, but we can change the chloroplast. Okay, we can get converted the chloroplast into chromoplast. So now let us see the chloroplast. Okay, so this chloroplast is again one of the important organelle of a cell. Now we have seen the chloroplast is found in a plant cell. Okay, we will not see chloroplast in the animal cell. So these are important for the photosynthesis process that takes place in the leaves. Okay, so the chloroplast which is there in the plant cell is a very important part, okay, or have a very important role in the photosynthesis process. So chlorophyll in chloroplast traps solar energy and it converts it into a chemical energy. So whatever solar energy is emitted by the sun, that solar energy is extracted, that solar energy is trapped by the chlorophyll, and it is converted into a chemical energy. And the stroma in the chloroplast contains enzymes, DNA, ribosomes, and carbohydrates that are necessary for photosynthesis. So these important materials, okay, or you can say them as genetic materials like DNA or ribosomes, okay, these important materials are there in the stroma of the chloroplast, and these materials are utilized in the process called photosynthesis. Okay, now here you can see this is the diagram of chloroplast. Stroma is there, then thylakoid, granum is there, there is one inner membrane and there is outer membrane. So by this we can say that chloroplast is double membrane structure, okay, and there is some intermembranous space within the two membranes, okay. So there is some space between inner membrane and outer membrane of the chloroplast. Now let us see the functions of the chloroplast. First function, chloroplast converts solar energy to chemical energy. Then second function, chromoplast give different colors to flowers and fruits. Okay. Then third, leucoplasts are involved in the synthesis and storage of food like starch, oils, and proteins. Okay. So these three types of plastids are very much important considering the development in the cell. Then mitochondria and plastids can produce proteins as well as replicate themselves as they have their own DNA and ribosomes. Okay. Now after studying the cell and cell organelles, you must have come to know that all the functions in the plant and animal cells are taking place smoothly due to the cell organelles. Such developed cells are called as eukaryotic cells. So we have studied about animal cell and plant cell in this chapter, and we have gone through the functions of different cell organelles, okay, and by this we have understood that the smooth functioning of the cell organelles has led to the development of the body of a particular living thing, okay, or living being. So whatever cells we have studied in this chapter, those are eukaryotic cells, okay, as they have developed nucleus, as they have developed organelles, okay. Last year you have studied prokaryotic cells of the bacteria. Now in this year we have studied the eukaryotic cells. Now let us now compare them. If you want to compare the prokaryotic and eukaryotic cells, how we can compare them, let us try to understand that. So here the differentiation or distinction between the two cells is given, okay. We will study the parameters of eukaryotic cell and also we will look into the prokaryotic cell. So the first thing, that the size of the eukaryotic cell is about 5 to 100 micrometer, whereas the size of prokaryotic cell ranges between 1 to 10 micrometer. The number of chromosomes present in eukaryotic cell is definitely more than 1, whereas only 1 chromosome is present inside a prokaryotic cell. Then nucleus, okay, definitely there is nuclear membrane, and inside the nucleus you will find nucleolus and nucleoplasm, okay, whereas in the prokaryotic cell you will find a nucleoid resembling nucleus. There is no true nucleus present in prokaryotic cell, okay. Then next distinction, that is mitochondria and plastids are present in eukaryotic cell, whereas they don't have membrane bound cell organelles. In prokaryotic cell you are not going to see any of the organelle which are membranely bound, okay. Examples present in highly evolved unicellular and multicellular plants and animals, okay. You will see that the unicellular organisms which are highly evolved like amoeba or euglena or paramecium, they are completely developed unicellular organisms, so they have a eukaryotic cell, and definitely all the multicellular plants and animals have eukaryotic cell, okay, whereas the example of prokaryotic cell is a bacteria. So some bacterias are belonging for belonging to this category, that is prokaryotic cell. Okay, so in this lecture we have completed our chapter, that is chapter number 10. We have gone through the different types of cell organelles, and we have also seen the functions. Okay, for this chapter, your homework will be you have to complete the exercise of this chapter. Okay, from the next lecture onward, we are going to start our new chapter. Okay, until then, goodbye.